Hello students, myself Dr. Mohammad Zafar Iqbal Nawalgund, Professor of Zoology, Department of Studies in Zoology, Government Science College Chitradurga. Today I am in the process of preparing e-content for the third semester BSc Zoology students affiliated to Davangere University. This content aims to align with the prescribed syllabus, offering comprehensive and accessible material to enhance student understanding at academic performance. And this semester has a paper titled as Animal Physiology and Biochemistry. And this paper has got four units are there. And out of 4 units, I am responsible for preparation of the e-content for the fourth unit, that is the metabolism. Dear students, as you know that the glucose is one of the most important and central molecule in the metabolic activities, and from where this glucose comes. And if you take the glucose, the most of the food what we take is in the form of glucose only, maybe either in the form of a starch, maybe in the form of a glycogen, maybe in the form of a sucrose. And if you take starch, the starch undergoes digestion by means of amylase. There are two types of amylases are there. One of the amylase is present in the salivary glands, and that is called the salivary alpha amylase. And one more amylase is present, and that particular amylase is synthesized and secreted by the pancreas. Hence, it is called as the that is the pancreatic alpha amylase. And this enzyme is responsible for cleaving the starch into maltose molecules. And whatever the maltose molecules and maltotrioses are there, these are going to be digested into the glucose by various other enzymes are all there. And in your physiology unit, you are going to study about in detail about the digestion process. And that's why I don't want to have a elaborated discussion on this particular topic. Just for your information that whatever the glucose molecules are there, these glucose molecules are generated in our body by means of the starch and glycogen. And these are the two very, very important type of the polysaccharides which are responsible for the generation of the glucose. And apart from that, whatever the fructose we take, that particular fructose also gets converted into the glucose. There are so many different type of the metabolic activities are there, and these metabolic activities are responsible for the conversion of the fructose into the glucose, and that's why the fructose also gets converted into the glucose. And in the same way, if you take the galactose is generally present in the milk, and whatever the galactose is there, the galactose also gets converted to the glucose. That's why if you take the glucose is nothing but it is a one of the very, very central molecule which is responsible for providing energy to the all the animals. This is one very, very important thing. Then if you take the glucose molecule, once the glucose is absorbed into the blood, then this particular glucose can undergo various type of the fates, actually pathways. It can enter into various metabolic pathways. One of the very, very important metabolic major pathway for the utilization of the glucose is nothing but it is the oxidation of the glucose by means of a glycolysis, and that is responsible for the conversion of the glucose into the pyruvate. This is the first major pathway which is going to be utilize the glucose. The second important pathway is nothing but it is to convert. If there is a already the person has got a sufficient energy, then that there is no need of the synthesis of the ATP molecule. Then whatever the excess glucose is there, that particular glucose is going to be stored in the form of a glycogen. That's why if you take the animals, in case of the animals, we store it in the form of a glycogen. And if it is an example of a plant, then it is stored in the form of a starch, or sometimes you can find that in case of the plants also you can find that it is also stored in the form of a sucrose. And this is the second major pathway of the glucose utilization. The third way of utilization of the glucose is nothing but it is the synthesis of the ribose 5-phosphate. And whatever this particular pathway is there, this particular pathway is called as the pentose phosphate pathway, that is called as the PPP. And whatever this particular pathway is there, this is also one of the very, very important pathway, because if you take this particular pathway is responsible for the synthesis of 5-carbon sugars, and it is also responsible for the synthesis of the NADPH, which is going to be utilized in the anabolic activity. That's why this is the third major pathway of the glucose utilization. And the fourth pathway for the glucose utilization is nothing but it is the formation of the extracellular matrix, and that we call it as a ECM. And whatever the ECM is there, we know that in case of the ECM, you can find that there are different type of the polysaccharides are present. And whatever these heteropolysaccharides are there, these heteropolysaccharides along with the proteins are responsible for the formation of the extracellular matrix. In this way, whatever the glucose which is taken by the organism is going to be utilized in either of these four major important pathways. In the next slide, here we are going to study how exactly the glucose is transported into the cell, because once it is absorbed, it will enter into the blood, and from the blood, that particular glucose must enter into the cell actually. And there are two types of the transporters are there, and these transporters are responsible for the transportation of the glucose from the blood into the cell. The first type of the transporters are nothing but they are called the GLUT. And whatever the GLUT is nothing but is a glucose transporters. And whatever the glucose transporters are there, these glucose transporters are nothing but they are the example of the facilitated diffusion. And this is the one type of the transporters which are responsible for the transportation of the glucose from the blood into the cell. The second type of the transporters are called as the secondary active transporters. That is the example, that is the SGLT, that is the sodium glucose linked transporters are there. And whatever these two transporters are there, these two transporters are mainly responsible for the transportation of the glucose from the blood into the cell. And if you take the GLUT, that is the glucose transporters, there are three different classes of the glucose transporters are there. They are called as the class 1 glucose transporters, class 2 glucose transporters, and this is the class 3 glucose transporters. And if you take the glucose, if the example of a class 1 glucose transporter is taken, then we have got some example, that is the GLUT1, then GLUT2, GLUT3, GLUT4. And apart from these GLUT4, we have got one more class 1, like one more example for the class 1, that is the GLUT14. And whatever these 5 transporters are there, these are the GLUT transporters which are responsible for the transportation of the glucose into the cell. And whatever these particular transporters are there, these glucose transporters are well studied. They have a high glucose affinity, and that's why they have the ability to transport very high quantity of the glucose from the blood into the cell actually. And out of that, the glucose GLUT4 is very important because it is insulin sensitive, that we are going to see in the next slide. And the second class of the GLUT transporters are called the class 2 GLUT transporters. And these are nothing but the examples are GLUT5, GLUT7, GLUT9, and GLUT11. And whatever these GLUTs are there, they are responsible for the specialized substances. Example, fructose. They are mainly responsible for the transportation of the fructose, especially if you take the example of a GLUT5, the GLUT5 is highly specialized for the transportation of the fructose from the blood into the cell. There is a class 3 type of the GLUT transporters are there, and they are nothing but they are the GLUT6, GLUT8, GLUT10, and GLUT12, and GLUT13. These are the intracellular. They have an intracellular role, and they are less understood, and that's why a lot of studies not been done on this particular thing. That's why we have a lot of information about the class 1 GLUT, and we have got sufficient information about the class 2 GLUT, and we have a very less information about the that is the class 3 transporters actually. And if you take the example of a different type of GLUT transporters, you will find that the GLUT1 is present in the RBCs, it is present in the brain, and it is present in the placenta. And whatever this particular GLUT1 is there, this is insulin insensitive. It means this particular GLUT transporter doesn't require it doesn't require the insulin, and even in the absence of the insulin, you can find that glucose can enter into the cell. And second important type of the GLUT transporters, they are called as the GLUT2. And in this way, you can find that GLUT it is present in the liver, it is present in the beta cells, it is present in the intestine, and it is also present in the kidney. And whatever these particular GLUT2s are there, the GLUT2s are glucose sensing, and they are bidirectional transporters. It means that glucose can enter into the cell, glucose can come out of the cell. That's why bidirectional movement of the glucose is possible with respect to GLUT2. And if the GLUT3 example is taken, the GLUT3 examples are transporters are generally present in the neurons, and they have got a very high affinity for the glucose. And glucose uptake is very important in case of the neuron. That's why they have a very high affinity for the uptake of the glucose by means of this particular transporter. The most important GLUT transporter is nothing but it is the GLUT4. And this GLUT4 transporter is present in the muscles, it is present in the adipose tissue, and whatever this particular GLUT4 transporter is there, this is insulin dependent. And whatever the uptake of the glucose is there, the uptake of the glucose depends upon the availability of the insulin. If the insulin is available, then only the glucose will enter into the cell. And if the insulin is not available, then the glucose is not going to enter into the cell actually. And that's why if the insulin deficiency will be there, then that particular person will suffer from the diabetes. And that's why whatever the glucose quantity is there, the glucose quantity starts building up in the blood, and that's why the transportation of the glucose becomes very less, and that results in the that causes the diabetes mellitus. And then the next important transporter, that is the GLUT5. These are the, as I have already told in the last slide, that they are present in the small intestine, they are also present in the sperm. And whatever these particular transporters are there, these transporters are mainly responsible for the transportation of the fructose, and they are not meant for the transportation of the glucose. They don't have the ability to transport the glucose, but they have the ability to transport the fructose. Then if you take the second type of the transporters, they are called as the SGLT. SGLT is a abbreviation, and the long form of this abbreviation is nothing but it is the sodium glucose linked transporters. That's why they are called as the SGLT. And if you take the SGLTs, there are two type of the SGLTs are there. One is called as the SGLT1, and second one is called the SGLT2. And whatever the SGLT1 is there, this is the transporters which are located in the intestine. They are also located in the kidney. And whatever these particular transporters are there, they are mainly responsible for the active uptake of the glucose and the galactose with the sodium. Here there is a exchange will take place. Sodium is going to be exchanged with the glucose molecule. That's why they are this type of the transporters are called the sodium glucose linked, because one molecule of glucose enters into the cell, and one sodium will come out of the cell actually. That's why they are called as the sodium glucose linked transporters. Here you will find that the sodium will come out of the cell, and there is an entry of the glucose molecule. That's why they are the they play this type of the role actually. The second type of the SGLTs are called as the SGLT2, and whatever the SGLT2s are there, these SGLT2s are generally present in case of the kidney. And whatever these particular transporters are there, these transporters are mainly responsible for the reabsorption of the filtered glucose actually. That's why whatever this particular glucose molecules which is present in the blood will enter into the cell either by means of the GLUT transporters or by means of the SGLT. That is a very, very important point for the students to remember. Most of the time what will happen, we may not be able to understand how exactly who is responsible for pumping the glucose into the cell, and that's why we have got a specialized type of the transporters are present in the form of the GLUT and the SGLT. And whatever these two types of the transporters are there, these two type of the transporters are mainly responsible for pumping the glucose from the blood into the this thing actually. And now if you take the oxidation of the okay, glucose, here we will find that the same thing. Here you can find that this is the fate. And in this slide, we are going to discuss about the oxidation of the glucose. I have already told in the earlier slide that whatever the glucose is there, the glucose get utilized by four different type of the major pathways. One of the major pathway which is responsible for the synthesis of the ATP or the generation of the ATP is nothing but it is called the oxidation or oxidation by means of the glycolysis, and this particular process is nothing but it is a glycolytic pathway. And in this slide, in this video, we are going to concentrate our discussion on the oxidation of the glucose by means of the uh, glycolysis. And if you take the glycolysis is nothing but it is a one of the very, very important metabolic pathway. And this particular pathway is not only present in case of the aerobic organisms, this particular pathway is also present in the anaerobic organism. And what is the meaning of the glycolysis? The glycolysis is nothing but it is a term which is taken from the which has got two different type of the terms which have been linked together. Glyco means sugar and lysis means breakdown, and that's why glycolysis means sugar breakdown. That's it. That is called the sugar breakdown, and that's why whatever this particular pathway is there, this particular pathway is responsible for the breakdown of the glucose into the pyruvate. And this particular pathway is also called as the Embden-Meyer of Parnas pathway, EMP pathway. Sometimes in your examination, instead of asking you to write a detailed discussion of the glycolysis, sometimes they may ask you to write about the EMP pathway. That time the students must be in a position to understand that EMP pathway is nothing but it is the glycolysis. Both terms can be used in alternatively. That's why this is the central role. This particular pathway plays a very, very important central role in the energy metabolism. And as I have already told you that whatever this particular glycolysis is there, this glycolysis is not only present in case of the anaerobic organism, it is also present in case of the aerobic organism. Why? Because this is a very ancient pathway. This particular pathway got evolved before the evolution of the oxygen on this particular atmosphere, and that's why this particular pathway is universal for all the organisms. You will find that the glycolysis is not only possible in case of the aerobic organism, the glycolysis is also possible in case of the anaerobic organisms. Okay, then here we are going to discuss about this particular pathway, some more information about the glycolytic pathway. This particular pathway takes place in case of the cytoplasm. Sometimes in your examination or in case of the viva, the examiner may ask you where exactly this particular pathway will take place, whether this particular pathway takes place in the cytoplasm or it will take place in case of the mitochondria. Then the student must be in a position to tell that whatever the glycolysis is there, the glycolysis occurs in the cytoplasm. Cytoplasm is the site where exactly the glycolysis is going to take place, and whatever the enzymes which are necessary for the glycolysis, all of them are present in the cytoplasm. And because of this reason, you can find that whatever this particular pathway is there, this particular pathway will take place only in case of the cytoplasm. And as I already told that it is not only possible in case of the aerobic and anaerobic, let's say, it is also possible. It is possible in case of the prokaryotes as well as the eukaryotes. And then whatever this particular pathway is, this particular pathway is mainly responsible for the conversion of the glucose into the pyruvate. And we already know that the glucose has got six carbon atoms, and these six carbon atoms will be converted into, it will be broken down into two pyruvates, and it means that one pyruvate will carry a 3 carbon atom. That's why 6 carbon will be converted into the 3 carbon containing compounds actually. And that's why whatever this particular pathway is there, this is the very important pathway. And pyruvate is one of the very important molecule. After once the pyruvate is formed, then you can find that various different type of the fates are possible with the pyruvate. But whatever before, in order to generate this particular pyruvate will find that the glycolysis is very important. And in the glycolysis, there are two phases are there. The first phase of the glycolysis is called the preparatory phase, and second phase of the glycolysis is called the payoff phase. Why we call it as a preparatory phase? In case of the preparatory phase, you can find that whatever the glucose molecule is there, the glucose molecule will undergo certain kind of modifications, and during this particular modification, you can find that it uses energy, and totally 2 ATP molecules will be consumed. That's why you can find out whatever this particular phase is there, this phase is not responsible for the generation of any ATP. Instead, this particular phase is responsible for the utilization of that is the ATP. That's why this is called the preparatory phase. The second phase of the glycolysis is called the payoff phase. Why it is called the payoff phase? Because in this particular 5 reactions, you can find that there is a production of the ATP molecules, and apart from the ATP molecules, you can find that this particular payoff phase is also responsible for the generation of the NADH. That's why both the ATP molecules as well as the NADH molecules will be generated in this particular payoff phase. That's why it is called the payoff phase. This is the overall reactions of the glycolysis. Here you can see that there are 10 reactions are present, and whatever these 10 reactions are there, these 10 reactions are responsible for the conversion of the glucose, 6 carbon containing glucose into the 3 carbon containing pyruvate. And in this way, you can find out whatever we are going to discuss individual reaction in detail. Just this is an overview of the total overall process of the glycolysis. And here you can see that whatever the glycolysis is there, there are 10 reactions are there, and there's a different type of the intermediates will be formed, and these intermediates will ultimately get converted into the pyruvate that we are going to discuss in the slides to come. Okay, the first reaction of the glycolysis is nothing but it is called the phosphorylation of the glucose, and this particular phosphorylation of the glucose is catalyzed by means of an enzyme called as the hexokinase. And this particular enzyme is responsible. Hexokinase in most of the cases, in sometimes you can find it in some of the tissues, there is a glucose kinase is also used. And whatever the hexokinases are there, these are the enzymes which are responsible for the transfer of the phosphoryl group from the ATP to the glucose molecule at carbon number 6. And that's why you find that at the end of this particular reaction, you can find that the glucose molecule will take a phosphate group from the ATP, and that is responsible for the formation of a glucose 6-phosphate. And after that, you can find that there is a one ADP molecule and one that is the proton will be formed. And whatever this particular reaction is requires energy. That's why in ordinary condition, this is a irreversible step. and only under specialized conditions, only by using a specialized type of the enzyme, that is the enzymes, it is possible to convert the glucose 6-phosphate into the glucose. Otherwise, you can find that whatever this particular reaction is there, this particular reaction is irreversible. And in this particular first reaction, there's a consumption of 1 ATP will take place. There's a 1 ATP molecule is converted into the ADP molecule. It means that there is a investment of 1 ATP, and that's why one phosphorylation reaction is responsible for the utilization or the consumption of 1 ATP. This is the first reaction. Here you can see that this is a glucose molecule. Here you can see that this is the carbon number 1, this is the carbon number 2, this is the carbon number 3, this is carbon number 4, this is carbon number 5, this is carbon number 6. And whatever this monophosphoryl group is there, the monophosphoryl group is going to be taken from the ATP. That is the, in case I think you might be knowing that there are 3 phosphoryl groups are present. That is the alpha phosphoryl group, beta phosphoryl group, and gamma phosphoryl groups are present. And whatever the hexokinase or the glucokinase are there, the glucokinase or the hexokinase are responsible for removing the gamma phosphoryl group from the ATP, and that particular phosphoryl group will be transferred to the glucose molecule at carbon number 6. That's why this particular, now you can see that the glucose is converted into the glucose 6-phosphate. And whatever this is what are very, very important. Why this particular phosphorylation of the glucose is important? If the phosphorylation of the glucose doesn't take place, then what will happen? Glucose can enter and go out of the cell at any time. That's why it is not going to be trapped. If once this particular glucose gets phosphorylated, then what will happen? The glucose gets completely trapped inside the cell actually. Now it carries the negative charge, and since it carries the negative, that's why the cell membrane will not allow the glucose 6-phosphate to go out of the cell. That's why the glucose gets entrapped inside the cell. That's a very, very important thing for this particular reaction. The second reaction is nothing but it's called the isomerization. Isomerization means one form is converted into the another form, and whatever this particular isomerization reaction is there, this is catalyzed by means of a enzyme called the phosphoglucoisomerase. And whatever this enzyme is there, this is the enzyme which is responsible for the conversion of the glucose 6-phosphate into the fructose 6-phosphate. And we know that glucose 6-phosphate, glucose as well as the fructose, both of them are 6 carbon containing compounds. And now what is happening? Only thing is that the carbonyl carbon is present at the carbon number 1 in case of the glucose, whereas in case of the fructose, you will find that the carbonyl carbon is present at the second carbon atom. And now what is going to happen? By means of the isomerization, you can find that the glucose 6-phosphate is going to be converted into the fructose 6-phosphate. And this particular reaction is a reversible reaction. At any point of time, the glucose 6-phosphate can be converted into the fructose 6-phosphate, and the fructose 6-phosphate can be again converted back into the that is the glucose 6-phosphate. That's why this particular reaction is called the reversible reaction. And in this reaction, you can see that this is the actual reaction. What happens? Chemical structures. Here you can see that this is the glucose 6-phosphate what we have got in the first reaction, and this particular glucose 6-phosphate will undergo isomerization to the fructose 6-phosphate. And here you can find that this particular, here you can find that the glucose is going to be converted into the fructose. But the problem is that it is not easy to convert the glucose 6-phosphate into the fructose 6-phosphate. That's why there's a sequence of reactions will take place. Usually, in the most of the time, you can find that the glucose 6-phosphate is present in a pyranose ring structure. This pyranose ring structure will undergo open chain form, and now once it gets converted to the open chain form, that then only you can find that this particular glucose 6-phosphate can be converted into the fructose 6-phosphate. And once this particular, this will also happen in the open chain form only. Once there is a conversion of the glucose 6-phosphate into the fructose 6-phosphate, again what will happen? It will get converted into the furanose ring structure. That is the fructose 6-phosphate will be formed, and whatever this fructose 6-phosphate is there, the fructose 6-phosphate has got a furanose structure. In this way, you can find that this is how this particular isomerization will take place, how exactly the glucose 6-phosphate will get converted to the fructose 6-phosphate. And the next important reaction is the second phosphorylation. In the first phosphorylation, already we discussed that the glucose gets phosphorylated to form the glucose 6-phosphate. And now in the third reaction, you can find that whatever this particular reaction is there, this particular reaction is very, very important, because here you can find that there's a second phosphorylation will occur, and that phosphorylation will be catalyzed by means of an enzyme called the phosphofructokinase 1, PFK1. And this particular enzyme is responsible for the addition of a transfer of the phosphoryl group from the ATP to the fructose. And here already we know that at carbon number 6 already there is a phosphoryl group is present. That's why whatever this particular second phosphate group is there, that particular phosphate group is going to be added at the carbon number 1. That's why whatever this particular end of this particular reaction, you can find that there is a formation of the fructose 1,6-bis phosphate is there, and ADP is will be produced because one phosphoryl group has already been donated to the fructose 6-bis phosphate. That's why I can find that there is a it will form the ADP, and there is a proton will be there. And whatever this particular reaction is there, this is a very important reaction, because this reaction is so important that there must be this is a rate limiting step actually. And whatever the regulation of the glycolysis is there, the regulation of the glycolysis mainly occurs at this particular point of time. That's why this is one of the very important step in case of the glycolysis. And now we'll see the that is the we'll see this particular next step, that is the cleavage. That is the fourth reaction, is nothing but is called as the cleavage. And this particular cleavage is nothing but it is the breaking up of the fructose 1,6-bis phosphate into the two compounds. One of the compound is going to be the glyceraldehyde 3-phosphate, and second compound is going to be the dihydroxyacetone. And that's why whatever these two sugars are there, both of them have got a 3 carbon atom. Glyceraldehyde 3-phosphate has got a 3 carbon atom, and at carbon number 3 you will find that there is a phosphoryl group is present. And in case of the dihydroxyacetone, there is a phosphoryl group is present, and that particular phosphoryl group is present at the carbon number 1. In this way, you can find that whatever the 6 carbon containing fructose 1,6-bis phosphate will be converted into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And one more important point I want to highlight here, that is we use two different type of the terms. One time, sometimes we use the word called as the bisphosphate, and in some other occasions we use the word called as the diphosphate. Bis means two, di also means two actually, but the context differs. In if you want to use the bisphosphate, whatever the phosphate groups are there, these phosphate groups must be present at a different locations. One of the phosphate group must be present in one particular position, and second phosphate group must be present on the some other location. Then only you will find that there's a this particular then only we can call it as a bisphosphate. And if you want to use the diphosphate, if you take the adenine ADP, here we use the word called diphosphate, because whatever these two phosphate groups are there, these two phosphate groups are connected to each other by means of a anhydride bond, and because of this reason you can find we use the word called the diphosphate. That's why the students must be very careful. Here you can't use the diphosphate, you must use the word called as bisphosphate, because one of the phosphate group is present at the carbon number 1, and second phosphate group is present at the carbon number 6, and in no way these two phosphate groups are linked to each other. That's why we must use the word called as the bisphosphate. We must not use the word called as the diphosphate. Okay, this is also reversible reaction. Whatever this particular reaction is there, anytime the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate can undergo condensation to form the fructose 1,6-bis phosphate. That's why it's a reversible reaction. The next here you can see that the next reaction is nothing but is the isomerization of the conversion of the glyceraldehyde 3-phosphate into the dihydroxyacetone phosphate. Here you can find that this particular reaction is catalyzed by means of an enzyme called as the triose phosphate isomerase, and this particular enzyme is responsible for the conversion of the glyceraldehyde into the dihydroxyacetone phosphate. And same way the dihydroxyphosphate can also be converted to, they are interconvertible actually. And here we know that whatever the glyceraldehyde 3-phosphate is there, this particular product is presented, it is directly on the path of the glycolysis, whereas if you take the dihydroxyacetone phosphate, you can find that it is not directly on the path of the glycol. That's why whatever the one molecule of the dihydroxyacetone phosphate must be converted to the glyceraldehyde phosphate. This particular reaction is a reversible reaction. And here you can see that, now you can see that this is the how exactly this particular interconversion will take place. Now whatever this particular, this is the glyceraldehyde 3-phosphate. Here you can see that the 3 carbon atoms are present, and whatever this particular phosphate group is present, the phosphate group is present in the carbon number 6. That's why it is called the glyceraldehyde 3-phosphate. And in case of the dihydroxyacetone phosphate, you can find that the phosphate group is present in the carbon number 1, and that's why you can find that this is the dihydroxyacetone phosphate. And whatever this particular dihydroxyacetone phosphate is there, it can be converted into the glyceraldehyde. That's why one molecule of the glucose is responsible for the generation of two molecules of the glyceraldehyde 3-phosphate, and henceforth whatever the products are, whatever the intermediates are there, all these intermediates will be 2 in number. That's why we are writing it as a 2x. It means that one of the glycerol, already we have got one glyceraldehyde, and after the conversion of the dihydrogen phosphate, you can find that there's a second glyceraldehyde molecule will be formed. That's why we have got a two glyceraldehydes, glyceraldehyde 3 phosphates will be there. Both of them will undergo glycolytic pathway, and hence they are responsible for the generation of two pyruvates in future. Okay, in the next reaction that is called the dehydrogenation, this particular reaction is catalyzed by means of an enzyme called the glyceraldehyde 3-phosphate dehydrogenase. It's a very complex enzyme. It has got three components. We call them as a E1, E2, and E3 components are there. And whatever this particular enzyme is there, this particular enzyme is responsible for the conversion. There's a dehydrogenation will take place, and that particular dehydrogenation is responsible for the conversion of the glyceraldehyde 3-phosphate into the 1,3-bisphosphoglycerate. And one very important thing in this particular reaction is that there is a one molecule of the NAD is used, and that is converted into the NADH. Whatever the dehydrogenation is there, two hydrogens are removed, and whatever these two hydrogens are there, one of the hydrogen will be transferred to the NAD, and that's why the NAD will get converted to the NADH, and this is a proton will be freed actually. And whatever this particular reaction is there, this is a irreversible reaction. That's why it is not possible to convert the 1,3-bisphosphoglycerate into the glyceraldehyde 3-phosphate. That's why this is another very important reaction which is required in the glycolysis. Now this is the how exactly the reaction will take place. This is the glyceraldehyde. Now here you can see that this is a glycerol 3-phosphate dehydrogenase enzyme is there, and now what will happen? NAD will get converted to the NADH. One of the hydrogen molecule which is removed from the glyceraldehyde 3-phosphate will be transferred to the NAD, and then this NAD plus will become the NADH, and one other phosphate group will be removed, and that particular phosphate will be in the form of a proton. That's why you can find that whatever this particular reaction is, the dehydrogenation reaction is there, this dehydrogenation reaction is responsible for the conversion of the glyceraldehyde 3-phosphate into the 1,3-bisphosphoglycerate. This is another reaction. Now there is the first this is the first step of the payoff phase. Here we are going to get the generation of the ATP. Now whatever the 1,3-bisphosphoglycerate is there, it has got a very high phosphoryl transfer potential, and that's why it wants to donate a phosphate group, and somebody is there to receive that particular phosphate. That is nothing but the ATP. That's why in order to get this stability, what this 1,3-bisphosphoglycerate will do, the 1,3-bisphosphoglycerate will donate a phosphate group to the ADP, and now we can find that ADP will get converted to the ATP, and whatever the bisphosphoglycerate is there, once one of the phosphate group is removed, automatically you can find that it will become the 3-phosphoglycerate. Now we'll see the reaction, how exactly this reaction will take place. Now here you can say that this is the 1,3-bisphosphoglycerate. Here you can see that one of the phosphate group is present at this carbon atom, and second phosphate group is present at the carbon number 3. Now what will happen? This is going to donate this particular carbon atom, that is the carbon, that is the phosphate group attached to the carbon number carbon number 1 will be donated, and that will be donated to the ADP, and once it receives that particular phosphate group, the ADP will get converted to the ATP. Now you can find that, since I already told you that one if you take the glyceraldehyde is present in two copies, one is directly in the form of a glyceraldehyde phosphate itself, the second one is the dihydroxyacetone phosphate, and dihydroxyacetone phosphate will also get converted to the glyceraldehyde phosphate. That's why two glyceraldehyde 3-phosphate will be there, and because of this you can find that already in this particular reaction you can find that two ATP molecules will be generated by utilizing two that is the 1,3-bisphosphoglycerate actually. And this particular reaction is catalyzed by means of an enzyme called the phosphoglycerate kinase, and this is the enzyme which is responsible for the transfer of the phosphoryl group from the 1,3-bisphosphoglycerate to the ADP molecule. The ADP will become the ATP. We'll move to the next slide. In this slide here, this is the next step, that is the 8th reaction, and this particular reaction is nothing but it is the transfer of the phosphate group from the carbon number 3 to carbon number 2. Again, now the 3-phosphoglycerate is a stable compound, and now it wants to produce, it wants to extract energy, and in order to make it further oxidable, what it will happen, this particular 3-phosphoglycerate will be converted into the 2-phosphoglycerate, and whatever this particular reaction is there, here what will happen, there is a transfer of the phosphate group from the carbon number 3. Here you can see that the phosphate group is attached to the carbon number 3, and now that particular phosphate group will be transferred to this particular carbon number 2. This is the carbon number 2. That's why now in this particular 2-phosphoglycerate, you can see that whatever the carbon atom which is present to the carbon number 3 is shifted to the carbon number 2, and that's why whatever this particular product is there, this particular product is called as the 2-phosphoglycerate, and this reaction is catalyzed by means of an enzyme called the phosphoglycerate mutase. This enzyme is responsible for the conversion of the 3-phosphoglycerate into the 2-phosphoglycerate. And the next reaction is the enolase. And this enolase, enolase enzyme, catalyzed by means of an enzyme called the enolase, and this is responsible for the conversion of the 2-phosphoglycerate into the PEP, that is the phosphoenol pyruvate. This is also called as the PEP, and the long form of the PEP is nothing but is the phosphoenol pyruvate. And whatever this product reaction is there, this is also irreversible reaction. It is not possible under ordinary condition to convert the phosphoenol pyruvate into the 2-phosphoglycerate, but you can convert this particular 2-phosphoglycerate into the phosphoenol pyruvate by means of an enzyme called as the enolase. And this is how exactly this will take place. Now here you can see that whatever the 2-phosphoglycerate is there, here there is a, this will undergo certain kind of modification, and that is responsible for the formation of the phosphoenol pyruvate. And here there is a removal of this particular oxygen actually. Okay, and that is responsible for the formation of the PEP. Okay, next, the last reaction of the this particular glycolysis is called as the that is the conversion of the phosphoenol pyruvate into the pyruvate. Here again you can find that this is the second example of the substrate level phosphorylation. And what will happen in this particular reaction? The whatever the phosphate group which is present in case of the PEP, that is the phosphoenol pyruvate, will be transferred to the ADP molecule. That once it receives a phosphate group, then automatically you can find that ADP will get converted to the ATP. And whatever this particular reaction is there, this particular reaction is going to be catalyzed by means of an enzyme called the pyruvate kinase. This enzyme is responsible for the conversion of the phosphoenol pyruvate into the pyruvate, and during this particular process you can find that ADP molecule will be converted to the ATP, and this is the irreversible reaction. Here this is exactly how exactly the reaction will take place. This is the phosphoenol pyruvate. Now whatever this phosphate group is there, it will be transferred to the ADP. The ADP will get converted to the ATP, and whatever this particular reaction is there, catalyzed by means of an enzyme called the pyruvate kinase. In this way, you can find that phosphoenol pyruvate will be converted to the pyruvate. Ultimately, you can find that by the end of the glycolysis, you can find that one molecule of the glucose is responsible for yielding the two molecules of the pyruvate, because one of the pyruvate is directly formed by means of the glycerol tetraphosphate, and second pyruvate is going to be formed by means of the that is the isomerization of the dihydroxyacetone phosphate into the glycerol phosphate. Actually, this is the how exactly these 10 reactions will take place. And now this is the one diagram. Here we can see that overall reaction that is the overall reaction of the glycolysis. One molecule of the glucose, two ADP molecules, two inorganic phosphate will be utilized, and two NADs will be utilized, and that is responsible for the formation of the 2 pyruvate and 2 ATP and 2 NADH plus 2 hydroprotons will be generated, and 2 molecules of the water will be generated. And whatever this product glucose molecule is there, the glucose molecules will enter into the cell, and glycolysis will take place in the cytoplasm, and there is an investment of 2 ATPs during this particular process in the preparatory phase. And ultimately at the end of this, by the completion of the glycolysis, you can find that 2 NADH molecules are synthesized, 2 pyruvate molecules are synthesized, and there is a generation of the 4 ATP molecules. But since we have already invested 2 ATP molecules in the preparatory phase, that's why there is a net generation of the net profit will be 2 ATP molecules. And that's why whatever these 2 NADH molecules and the 2 pyruvates are there, if there is a oxygen is available, then what will happen? They will enter into the mitochondria, and if the oxygen is not available, then they will go through another pathway, other fate, and that we are going to discuss in the next that is the video. And for the time being, just remember that depending upon this particular condition, just concentrate on the glycolysis. The glycolysis will take place in case of the cytoplasm. A molecule of the glucose will be utilized, and that will be oxidized into the 2 molecules of the pyruvate. 2 ATP molecules will be consumed in the preparatory phase, and that is responsible for the generation of 4 ATP molecules and 2 NADH. Since we already invested 2 ATP molecules, that's why there is a generation of 2 net ATP molecules will be generated. Okay, this is the energetics of the glycolysis. Here I have given some of the reactions where exactly there is an investment of the ATP will take place, and where exactly we are going to get the benefit. In the first reaction itself, that is the glucose will get converted to the glucose 6-phosphate. There is a phosphorylation take place, and during that particular phosphorylation, you can find that the ATP is the donor of the phosphate group. That's why one ATP molecule is considered. That's why minus one ATP. And in the second reaction, you can find that in the second phosphorylation reaction, where you will find that the fructose 6-phosphate will be converted into the fructose 1,6-bisphosphate. And here also you can find that there is another phosphate group is added to the fructose, and that particular phosphate group is going to be added at the carbon number 1. And the donor of this particular phosphate group is nothing but it is the ATP, and that particular ATP molecule will be converted to the ADP. In this way, you can find that here in the preparatory phase, you can find that 2 ATP molecules are invested, and this investment of the 2 ATP molecules will take place. Now, in the payoff phase, here you can find out the first payoff in the first benefit of the payoff phase is that whatever the glycerol tetraphosphate is there, it is responsible for the dehydrogenation reaction, and that is responsible for the transfer of that is the hydrogen to the NAD plus, and that's why the NAD will convert into the NADH, and whatever these NADH are, they are going to be used in the future for the generation of the ATP if it undergoes oxidative phosphorylation. That's why this is nothing but it is a gain of 2 NADH molecules will be generated. And the second important the reaction which is responsible for the generation of the 2 ATP in a molecule is nothing but is the conversion of the 2,1,3-bisphosphoglycerate into 3-phosphoglycerate. And during this particular process, you can find that 2 ATP molecules will be converted into the 2 ATP molecules, and that's why there is a gain of 2 ATP will be there. And in the last reaction, that is the 2-PEP, that is the phosphoenol pyruvate, will be converted into the pyruvate. And during this particular process, you can find that one phosphoryl group will be transferred to the ADP, and that's why 2 ADP molecules will be get converted to the 2 ATP. Altogether, you can find that if you want to see what is the net gain or what is the net ATP and NADH production, then we can say that at the end of the glycolysis, you can find that there is a net generation of the 2 ATPs will be there, and there is a net generation of the 2 NADH will be there. This is the net, this is the energetics of the glycolysis. Okay, and how exactly the next important thing is the regulation of the glycolysis. The first important point for the regulation of the glycolysis is nothing but is the availability of the substrate. For the glycolysis to happen, there must be availability of the glucose. If the glucose is present, then only there is a glycolysis will take place. And if the product, if the substrate is not available, then there is no possibility of the glycolysis. That's why this is the first way by which the regulation of the glycolysis will take place. The second regulation is nothing but it's called the allosteric regulation, that is the enzyme regulation. This we are going to discuss in the next slide. And the third type of the regulation is nothing but it's called the hormonal regulation. Altogether, you can find that there are 3 levels of the regulations are there. The first level of regulation is nothing but it is the substrate availability. The second level of regulation is the allosteric regulation by use of the enzymes. The third type of the regulation is nothing but is the hormonal regulation. Okay, in the first type of the in the second type of that is the regulatory enzyme. There are 3 important of the regulatory enzymes. Allosteric regulations are there. One is with respect to the hexokinase or the glucokinase, and these enzymes are responsible for the conversion of the glucose into glucose 6-phosphate. And whatever the ATP molecule is used, and this particular reaction is responsible for trapping the glucose into the cell. Okay, the second reaction is the phosphofructokinase 1, and this enzyme is responsible for the conversion of the fructose 6-phosphate into the fructose 1,6-bisphosphate, and it is a rate limiting enzyme, and it is a step where exactly the maximum regulation of the glycolysis will take place. The third point of regulation of the enzyme is the pyruvate kinase, and whatever the phosphoenol pyruvate is there, it will get converted to the pyruvate, and during this particular process you can find that there is a possibility of the regulation of this thing will be there. In case of the hormonal regulation, you can find that there are 3 important type of the hormones are there. One of the important hormone which is responsible for the which plays a significant role in the glycolysis is nothing but the insulin. And what insulin will do? The insulin is responsible for the activation of the glycolysis in the liver. And whatever this particular insulin is there, it increases the expression of the glucokinase. It increases the concentration of the phosphofructokinase 1, and it also increases the concentration of the pyruvate kinase 1. And the next one, the next important enzyme is the glucagon. And you know that glucagon works opposite of the insulin. And whatever the glucagon is there, it is inhibited. It inhibits the glycolysis in the liver. What it does, it inhibits the glycolysis. And whatever this particular glucagon is there, it actually inhibited in the by means of the fructose 2,6 bisphosphate and activates the that is the pKa, and in this the next reaction, the next enzyme is the epinephrine. In this enzyme we will find that that is also another enzyme which plays a significant role in the regulation of the glycolysis. Okay, these are the some of the important points related to the disorders related to the glycolysis. Already we know that diabetes mellitus is directly related to the insulin. Whenever a person suffers from scarcity of the insulin, that is responsible for the diabetes mellitus. Here we can find that insulin is responsible for the, if there is a decrease in the titer of the insulin, that decreases the glycolysis in the liver, and there is a Warburg effect. In the Warburg effect, what will happen? Even in the presence of the oxygen, the cells will prefer the glycolysis. They never go for the oxidative phosphorylation. That thing is called as the Warburg effect. Or it is very common in case of the malignant cells. And the third one is the pyruvate kinase deficiency. This is effect is that there is a hemolytic anemia due to the impaired ATP production will be there, and this is the overall cycle. All reactions we have studied individually, and here whatever this particular picture is there, this particular image will give a complete overall idea about the how exactly the glycolysis will take place from the reaction 1 to reaction 10. And during this particular process, altogether you can find that 2 NADH molecules will be synthesized and 4 ATP molecules will be synthesized. But in the preparatory phase, since already 2 ATP molecules have been invested, that's why there is a net production of only 2 ATP will be there. Okay, for this I have referred some of the books, that is the Lineage's Biochemistry, Strayer's Biochemistry, and Satyanarayana Biochemistry. Students are strictly advised to refer to the books for more information and knowledge. Thank you very much. In the next slide, we are going to discuss about the fates of the pyruvate. Thank you.